Hopefully I'm now sharing my main screen.
Now we are.
Great. Thank you. So thank you very much for having INOVIQ present today. I'm going to walk you through the company and our key programs. So INOVIQ is a company that's developing next generation exosome capture tools and precision diagnostics to improve the diagnosis and treatment of cancer. We've got a number of patented technology platforms for exosome isolation and biomarker detection. We have products in market for exosome research and detection of bladder cancer, and we have a multi-product pipeline for cancer diagnostics with some compelling early data in both breast and ovarian cancer. In terms of the company, we're currently sitting at around a $58 million market capitalization. We've got an experienced board and management team. Our chairman is Dr. Jeff Cumming. I'm the Chief Executive Officer, and I also have Dr. Greg Rice as our Chief Scientific Officer, who's got considerable experience in diagnostic development and commercialization, including in exosome-based diagnostics. We have a broad pipeline, mostly focused at the moment on breast and ovarian cancer, both screening and monitoring applications. This gives us multiple shots on goal for our development pipeline, which is at various stages. Just a quick introduction of the difference between screening and monitoring. Screening is for detection of disease in asymptomatic people before they have any symptoms, so it's before diagnosis. And then our other type of tests are monitoring tests, which is after people have been diagnosed with disease, and we're monitoring both treatment response and disease recurrence. So one of our first target markets is ovarian cancer. Ovarian cancer is the 8th most common cancer in women and the #1 cause of gynecological cancer deaths. The problem with ovarian cancer is that there is no tests that are currently available for screening of ovarian cancer in what we call average risk asymptomatic women. That means that the majority of women are actually diagnosed after they've got symptoms in the late stages, and that means that only 25% of women will survive for 5 years. If we can detect ovarian cancer in the early stages, which is stage 1 and stage 2, before the cancer is spread beyond the pelvis, that then can improve women's chance of survival to over 90%, which obviously is a fantastic outcome. So we're focusing on developing a screening test for ovarian cancer based on exosomes, which meets an unmet need in this area. The only blood test that's currently available for ovarian cancer is called the CA-125 test, which is a blood test for monitoring ovarian cancer. So that's after the disease has been detected. And essentially this test does not pick up early stage ovarian cancer. So you can see that less than 50% of ovarian cancer is picked up by the CA-125 test. So it is really a test for late stage disease. We are developing a test to improve on the CA-125 test, which I'll speak about in a moment. So firstly, our ovarian cancer screening test we have in development with the University of Queensland. This test is actually an exosomal based test. So what that means is the biomarkers in that test come from exosomes. There's multiple biomarkers, and the original work from the University of Queensland looked at 7 different microRNA and protein biomarkers. They were combined in an algorithm that was then tested in over 450 samples, and that showed over 90% detection of early stage ovarian cancer. So that's stages 1 and 2. So you can see here that there's a very clear detection of the cases compared to the controls. So that's an outstanding result. So we're now working with the University of Queensland to develop that test, and we have an exclusive option to license the development and commercialization rights to that test. This page really shows that we're currently doing this, what we call the Ovarian Cancer 97 study. That was a study to prove that ExoNet is fit for purpose to develop and commercialize this exosome-based ovarian cancer test. We expect to report data on that study very soon, in fact, in December. So watch the space. Once that study is completed, so in early next year, we will be completing a study called the Ovarian Cancer 250 study. What we're looking to do there is to show bioequivalence between the results we got from this study and between plasma, because this study was done in plasma samples compared to serum. Why that's important is that the world's largest biobank for ovarian cancer samples came from this UK CTOC study, where they collected samples from over 200,000 women over a 15-year period, and that was all serum samples. So in order to access those samples, we need to show bioequivalence so that we can then run a study in about 3,000 samples to clinically validate this test. In terms of upcoming data points, we expect to report on initially the analytical validation study by the end of the first half of next year, so in June, and then on the larger clinical validation study before the end of 2023. Moving on to breast cancer, John's already gone extensively through the breast cancer background, so I'll just say it is the #1 most common cancer in the world and the #1 cause of cancer-related deaths in women. Again, as John said, there is a screening test available for breast cancer, which is a mammogram, which, depending on the country, is recommended for women anywhere from 45 years of age onwards. We're currently focused on improving on the approved monitoring test called CA15-3, which detects treatment response and recurrence in women that have already been diagnosed with breast cancer. This CA15-3 test here shows that it can't detect its detection of early stage disease is around 60%, whereas it can detect about 77% of women in late stage disease. We're looking to improve on that test with our sub-B2M breast cancer monitoring test. So, having a look at our breast cancer monitoring test, that's currently in development with a contract research organisation in the United States called ResearchDx. We have commenced an analytical validation study of the sub-B2M assay, which is looking to improve on the existing CA15-3 test. The interim data shows that our assay can detect both early and late stage disease. The next steps is to actually establish reproducibility of this assay. And then at the end of that, we expect to be able to report on that data within the next month or so. Following that, we will be doing a breast cancer monitoring study. The breast cancer monitoring study is looking to show that our INOVIQ test is better than the CA15-3 test, so that we improve sensitivity and possibly specificity of that test. So sensitivity being the ability to detect breast cancer, and specificity is showing that if somebody doesn't have disease, then they get a negative test result. So moving on to one of our other areas is a sub-B2M SPR test. The original work by Griffith University was actually done using what's called SPR instrumentation. That instruments are not used in commercial labs around the globe. However, we have recently signed an agreement with a company called Nicoya, based in Canada, that have developed a benchtop instrument that could be used in commercial labs worldwide using this SPR technique, which is extremely sensitive for detection of a range of biomolecules, including New5GC, which is the sugar we're interested in, that's found on a range of cancer cells, which include breast, ovarian, prostate, and a range of other cancers. The original results using SPR showed that this sugar can be detected across all stages of breast and ovarian cancer with over 95% sensitivity and no false positives. This was a case control study. We're currently transferring this SPR test to Nicoya's platform called the Alto, and that's about a 5-month program, so we'll expect to report data on that sometime in the first half of next year. This test will initially be developed as a what we call a cancer risk assessment tool, because it does detect NEW5GC across multiple cancers. So it could be included as a routine blood test that showed if there was an elevated NEW5GC test, that would be an indicator that you are at higher risk of having cancer and should go back for further work. Down the track, there is potential to further develop this test as a multi-cancer detection test. That would be a two-step process where if you got a positive NEW5GC test, then we would have to follow up with a further test looking at a range of different potentially antibodies to show which tissue it came from, i.e. whether it was breast, ovarian, prostate, pancreatic, etc. Just touching on the commercialization strategy, our strategy is to enter the US market first as a laboratory developed test. We need to do that with a laboratory partner. That partner has to operate a CLIA accredited laboratory. We're currently working with ResearchDx, which does actually have a CLIA accredited laboratory. As John actually mentioned, the majority of tests in the United States are developed as LDTs rather than as IVDs. However, we may move forward into an IV to get an IVD clearance or approval later, but that would involve a larger scale clinical study, but that would enable us to distribute kits to multiple labs within the USA. Once we've achieved success in the US market, we also intend to develop the test in other markets, including Europe, Australia, and Asia. Just touching on the market potential, these are very large global markets. If I'm looking at the USA, the 5 major European markets in Australia, there's over 120 women that are over the age of 50 up to 79. So I think I said 120 million women that could be that are eligible for these types of screening tests. That equates to about an $18 billion market, whether it's breast or ovarian cancer, based on a test price of around $150 per test. Beyond that, the monitoring market, which is in women already diagnosed with disease, these are smaller markets, particularly in ovarian cancer, because that's not a very prevalent disease. It's number 8, as I said, in women, whereas breast cancer is actually the most prevalent disease in the world, and that's about a $670 million market for monitoring alone. These, on the left-hand side of the screen, you can see that the current ovarian cancer market is about a $1.9 billion market. That's mostly made up of monitoring tests and which includes basically imaging tests and blood tests for CA-125. And on the breast cancer side, that's both covers screening, monitoring, and other types of diagnostic tests. I'm just going to briefly touch on exosomes. Reason being is that we have a exosome capture tool for isolating exosomes from a range of biofluids. Essentially, these exosomes are small vesicles that are released from most cells in the body. They contain a range of biomolecules such as DNA, RNA, proteins, and others that we can use to develop diagnostic and potentially therapeutic applications for a range of diseases such as cancer, metabolic disease, inflammatory, and neurodegenerative diseases. Our ExoNet product is a pan-exosome capture tool, meaning that we're capturing exosomes from multiple tissues in the body. That product is now available both online around the world, but also we've launched it officially in the USA with a partner called Percorso Life Sciences. We have a contract sales team in place there, and Percorso provide logistics services direct to clients, which include universities and research institutes, which are developing a range of exosome-based diagnostics and therapeutics across a range of different areas. The sales team has actually launched its first sales campaign to over 1,000 researchers in the United States, and we expect that results will be delivered based on that in the first half of next year. The other important point is that we're actually expanding the pipeline of these ExoNet capture tools. And this is based on some of the collaborations that we have with various researchers in the cancer and neurodegenerative space, where they're interested in specific exosome capture tools for brain-derived EVs, which we call NeuroNet. And also a specific capture tool for really enriching for cancer-derived EVs, which we call TexoNet. So again, watch this space in the new year for these tests. ExoNet's got important advantages in terms of we can capture exosomes within 15 minutes. This can compare to other capture methods that are routinely used in laboratories around the world that can take as long as 2 to 12 hours to capture exosomes from samples. This is particularly important for developing and commercializing diagnostics that are going to be used in laboratories around the world. And that's why we're also developing automated solutions for ExoNet that'll enable high throughput processing of samples for exosome-based diagnostics. And again, watch this space next year for this area. We have performed studies that show that ExoNet outperforms 4 other commercial kits that are available on the market by some of the largest players in the exosome space. This graph here is a bit counterintuitive. The lower CT values here, this is ExoNet in the bright blue, shows that it outperforms the other kits. So high is bad and low is good, and ExoNet clearly outperforms the other kits, or at the very least is equivalent. Just finishing up, we had a big year during the year. Essentially, we contracted ResearchDx to develop and commercialise our sub-B2M tests. We signed an agreement with Nicoya in order to develop a sub-B2M SBR test. And we signed our first agreement with the University of Queensland to develop an ovarian cancer screening test based on exosomes that utilises our ExoNet product for capturing those exosomes. On top of that, we have launched our ExoNet product in the United States with a sales team, and look forward to seeing revenues generated next year in relation to our ExoNet sales. Coming up, most importantly, you can expect to see results from our sub-B2M breast cancer immunoassay. As I said, the analytical validation is underway in the lab. We expect to report on data showing accuracy across the various stages, which is stages 1, 2, 3, and 4 for breast cancer. And we will be performing a clinical study in the new year comparing the sub-B2M breast cancer test to the CA15-3 test for monitoring, and expect to be reporting on that during the year, followed by transferring that test to a laboratory partner, which we expect to be ResearchDx's clinical lab called PacificDx, so that the test can then be offered for sale towards the end of 2023. On the ExoNet side, as I said, we will be launching new products for brain-derived exosomes and cancer-derived exosomes in 2023. We expect to announce results on our exosome ovarian cancer screening test with UQ. In fact, we expect to announce data from the initial work in this month, followed by results in both June and then December of next year. And most importantly, we expect to launch the sub-B2M immunoassays as laboratory-developed tests in the States by the end of next year. Thank you very much.
Thank you. A lot to cover. We covered a lot of ground there. Just a really quick question in the interest of time. How does the data need to look to be able to commercialise successfully the sub-B2M monitoring tests?
Thanks, Chris. Essentially what we need to do, so with the breast cancer monitoring test, we need to show that the sub-B2M breast cancer test outperforms the CA15-3 test, which shows about 75% sensitivity for detection of late-stage disease and about 80% specificity. So we need to be able to have a better result in terms of sensitivity and specificity for our breast cancer monitoring test. And that's what our clinical study has been designed to do.
And effectively, as you nicely earlier, the data in the university test in the lead-up have been compelling. So if you can reproduce that's a very strong valuation, that clinical value proposition.
Indeed it is. Just pointing out the tests, the results from Griffith University were based on the SBR platform. So that's the new test that we're developing, which is the sub-B2M SBR test. So that test just detects new 5GC. It doesn't detect the monitoring test is specifically for detecting the CA15-3 molecule that's conjugated with new 5GC. Hence, it's used for monitoring applications as opposed to earlier detection.
Understood. No, that's great. Thanks very much, Leanne. That's been Dr. Leanne Hinch, CEO of INOVIQ. Thank you.
Thank you very much.